Like take advantage of the really, really broadcasting our
Yeah, no, that's great.
right? No reason not doing that.
I know.
Ready to get started top of the hour in 2 minutes?
Okay.
Yeah, let's just start.
Okay.
Okay. All right, let's get started. My name is Ed Guo. I'm the chair of the Department of Biomedical Engineering at Columbia University here. On behalf of the department, and then I'd like to welcome all the new students and also continuing students. This is the biggest class in the department history. And we're also so glad we do in person now. I think there's some echoes in here. So good to see your face and continue these traditions in here. So now I'm going to go back to my script. So today we continue to offer this lecture in a hybrid format for both the virtual and the live audience. Our speaker will present approximately 40 to 45 minutes, and then we'll leave about 15 minutes at the end for questions and answer. So for our virtual audience, throughout the presentation, please use the Q&A feature. I'm pretty sure everybody has now using Zoom quite a lot, using the Q&A feature, but not the chat to submit your questions. We will try to answer as many questions as possible during the Q&A sessions at the end. For the live audience, you can also have a chance to ask a question during the Q&A session as well. Okay, so today we're very excited to welcome our own Professor Treena Arinzeh, and Treena joined us this summer as our newest professor of biomedical engineering at Columbia University. I'm very, very pleased, and we're able to recruit in Treena, cross the river to this Manhattan. So she was a project manager at the stem cell technology company, or Series Therapeutics in company, and then joined the faculty of the New Jersey Institute of Technology as one of the founding faculty member of the Department of Biomedical Engineering there. He served as an interim chairperson and also graduate director. Treena has been recognized for numerous awards for his research, including the Presidential Early Career Award for Scientist and Engineer, called PKs. For those young students don't know what that one is, actually you go to White House and receive the awards from the president. So she's also fellow of American Institute of Medical and Biological Engineering, Biomedical Engineering Society, National Academy Inventors. He has been recognized for her efforts as diversity and inclusion as well. She's currently director of diversity of the National Science Foundation Science Technology Center for Engineering Mechanobiology, which is a multi-institution center led by University of Pennsylvania, as well as Columbia now. Welcome, Treena.
Okay. Can you hear me? Yes, you can. The microphone's working. Very good. Well, welcome. all. Yes, I'm brand new here. Came about a few weeks ago, moved in. And so I'm happy to be here. And so I'm looking forward to talking to you all about some of my research. And I wanted to just make a couple of announcements. I wanted to make a couple of announcements while I'm sharing. Because I wear a few hats. And so this year I am co-chairing the upcoming Biomedical Engineering Annual Society, the Biomedical Society Annual Meeting. So some of you hopefully are joining. There we go. So hopefully some of you guys are going to San Antonio. It's a great opportunity to network, learn about graduate school, postdocs, doctoral divisions, and things like that. For senior guys. And then also, as I said, as Dr. Dowd mentioned, I'm also a part of the Center for Engineering and Nanobiology, which is a consortium of universities, and Columbia is now a part of that. And so we offer, again, there's some undergraduates here. We offer a summer internship for rising juniors and rising seniors. So please look out for that. Applications are coming, are due soon. I think so. We're still checking.
Yes, sir.
Okay, I'm using this now. Okay, sorry about that. All right, so I just wanted to make those announcements. All right, so the research. So my area of research is, again, tissue engineering, regenerative medicine. Again, you all should be pretty familiar with this. And again, here's just kind of a schematic of the kind of conventional approach that is used where we're using cells, body's own cells, a patient's own cells, or maybe donor cells from another patient. And they're used as building blocks to repair or regenerate tissues. But the key here and what my lab focuses on is what's shown number 3, that scaffold material. So we're designing scaffolds or these biomaterials well in terms of their physical properties, their chemical material properties, in order to provide the appropriate cues to those cells. So again, these cells can be stem cells or other cell types. My lab has focused primarily on stem cells. And so we may use them in combination with those scaffolds as a therapy, or we've been now designing some of our biomaterials such that we're using those stem cells as models to screen and develop our biomaterials. So those biomaterials can be implanted by themselves into the body and coax the body's own cells to regenerate. So there's a number of approaches. And so I'll try to just cover some of those. So we focused a lot of our work in the area of these adult stem cell derived or stem cells that are derived from adult tissue. Mainly we're interested in these because they're well characterized. For decades, the ones that come from the bone marrow. And so again, the ones that are coming from adult tissue, although they are rare in number in these tissues, if you can isolate them, culture them, they can grow up to fairly large numbers. And so, like I said, they've been characterized, they've been shown to be safe, the ones that come from the bone marrow, and we're using these mesenchymal stem cells, the ones that will give rise to connective tissues. Again, our interests are mainly in orthopedics and the bone and cartilage world, but we also are in neural tissue regeneration. And I'm gonna talk about our efforts in those different applications. Okay, so I'll start first in bone, because our earliest work, and just to kind of highlight a couple of those things, and really the rationale behind why we are using this approach of the stem cells, either as a model or in combination with our materials. So I'll just highlight some of those studies in the area of bone, and then I'll move into these other applications, which are kind of more of our efforts as of late. So why do we use them for bone? Why do we use or need this approach for bone? It's really for these large defects that would not ordinarily heal on their own. So these may be due to trauma. They may be due to cancer that's present in the bone and that has to be removed. Or spinal fusions, right? Where you have this kind of degeneration of the disc and this is a way to stabilize. And so bone grafts are used for that, but they're not adequate, right? You usually are using very large amounts of these allografts, and they don't provide the necessary biological cues to really rebuild healthy bone. So it would be the approach, again, the scaffolds with the stem cells. And so our earlier work established that we can develop these bioceramics, these hydroxyapatite tricalcium phosphate porous ceramics that work well with these MSCs or stem cells, and that they allow for the attachment of these cells. If you notice the texture of these scaffolds, they have very kind of nano topography or porous topography, as well as these very large pores to allow for bone ingrowth and infiltration. But it's that nanostructure on the surface that allows for those cells to attach and differentiate. And so we've shown in models then, in particular this one, that we can use donor cells, these are allogeneic cells, or even, again, the autologous cells to repair these large defects. So shown here are x-rays. Again, if you treat it, if you leave the bone untreated, you're left with this huge defect, right? Empty defect. But if you treat it with that combination of cells, you get this bridging of the defect. And so you follow that with a number of different, outcomes of histology and we had to look for whether the cells were present. And so we were able to show that those cells were present in those tissues, building the bone tissue along with integrated with the scaffold over time. So this really served the basis then for FDA approval for the use of these donor cells for a number of clinical trials. Again, establishing that they're safe and efficacious in particular for bone, but there are other studies and for other applications. And there's currently a product on the market that utilizes this approach, this OsteoCell Plus. So lessons learned from this is that's great. We were able to demonstrate that we can heal these bone defects. But that original biomaterial actually didn't go forward into that product, right? So the OsteoCell that's on the market right now is those allogeneic stem cells, but with an allograft. So we kind of went back to like this allograft still material that we really don't like. Why is that? Because that bioceramic, even though great in the animal models that we showed, it is brittle, okay? And it didn't resorb. So the orthopedic surgeon said, no way, we can't use it in weight-bearing defect sites. It's going to be prone to fracture. It's going to stay there, and it's brittle, right? So what do? We look at materials then that can be designed better, that have improved mechanical properties, but still have that property of inducing those cells or promoting those cells to form bone tissue. And so we've focused a lot in my laboratory on the design, the architecture of those materials that we're using. So you can even have, again, that kind of porous block-looking material where the cells are really small in comparison to the structure or size of those pores, or you can create features that are much smaller in scale, right? And so again, looking at a more fiber structure is what we focused on in my lab, so you can really tailor the small micron to nano features of those materials. And what that does is allow for the presentation of more proteins, right? More factors to those cells. Cells will tend to adhere, and then it promotes their function as you get smaller in scale in those features. So we use this process of electrospinning, which is shown here. Other labs use it. We've been able to really optimize this process such that we can control in those fiber dimensions, as well as orientation of fibers. And so it's a fairly simple process. Again, where you're applying this high voltage to your material of interest, polymer that may be dissolved, or composites that are dissolved in some solvent. That solvent, as it is being ejected, or polymer solution as it's being ejected at a certain flow rate, is charged, and it results in the solvent being evaporated, and you get these fibers. And these fibers can be, again, depending on parameters used, we can control dimensions on that. So small nanometer up to fairly large micron scale dimensions, as well as orientation. So we've shown in a number of studies that it does affect cell adhesion. And so again, if you get down to that small scale, micron scale, You can really enhance cell adhesion as shown here. This is just some of the actin filaments, as well as we looked at focal adhesion kinase expression as an indicator of integrin, this integrin adhesion. We see that's expressed really high, and you get really organized integrin, these integrin filaments. So when we return back to the bone application, I'll just highlight a couple of things here and finish this up. So we went back to the bone, right, application. We said, okay, what are we going to use here? So we wanted big fibers, big interfiber spacing, right? The big pore space, because we need bone and blood vessels to fill in there over time, right? But we still want some of that nano features because we like that good cell adhesion as well. And then we want to incorporate that bioceramic because we know that calcium phosphate-based material promotes that stem cell differentiation. So we created this fiber composite, polymer and ceramic. Ceramics are built into the fibers with these nano ceramics. And we've been able to demonstrate now in a number of different models. This just is in the sheep animal model that we're getting pretty robust bone formation. And this is using the material by itself or in combination with bone marrow. So we can load bone marrow has those stem cells, and we also get this robust bone formation or integration into those defects. We also get that resorption of the biomaterial, and that's important. So again, over time, by 16 weeks, we get that degradation. And so again, that's important for remodeling of the bone. And so we're currently now investigating that, again, as a product for FDA clearance through this 510K mechanism. Okay, so I wanted to highlight some of those things where we are kind of pretty advanced in the bone area. Now I'm talking about cartilage, some of our latest technology that we're working on in cartilage, and then I'll get into the neural tissue engineering. And surprisingly, they're related. And I'll try to touch on that in the development of our biomaterials. And so we're kind of using similar materials in both applications. And so cartilage is a big area of interest. Osteoarthritis is a major problem, affects millions of people in the United States and worldwide. And there's no current technology out there, right, that will fully regenerate functional cartilage, meaning that you apply load to it, you're not going to feel pain, right? Because once you tear that cartilage, usually what fills in is fibrocartilage, fibrous cartilage. So not quite the healthy stuff that you want. So we go back to our biomaterial design. We first think about our fibers and what dimensions that we want. So we identify, again, big fibers for this, because when we put our stem cells on those scaffolds, what we find is, again, for them to become chondrocytes, unlike becoming bone cells in this case, for them to become chondrocytes, they need to attach to one another, form these aggregates, okay? So they still need to have some adhesion to the scaffold itself, but they do need to be able to have cell-cell interaction. So shown here, these aggregates on the far right, those are really only seen or promoted on the materials that had the larger micron sized fibers. So 5 and 10 microns in size, that PLA3, PLA4. So we use that design then going forward, but we are now getting into kind of the more, I call interesting compositions, meaning that they will, again, provide additional cues to those cells to drive them to become chondrocytes, those stem cells to become chondrocyte. So we look at the native cartilage tissue, and we look at it in development as well as, as adult. But what's clear in the cartilage tissue is that, yes, you've got fibers just like our scaffolds, which is great. You've got collagen type 2 fibers. But what's decorated on those collagen type 2 fibers is these glycosaminoglycans. So there's different collagens, and those collagens actually bind to glycosaminoglycans. And what's well known is that there's chondroitin sulfate, and there's some other types of GAGs that are present. And people have looked at those materials. And so we can design our materials to have that chondroitin sulfate, but we chose to go one step further than that. So we've developed these biomimetics or bio-inspired materials to mimic that GAG, okay, that glycosaminoglycan. So shown here out of cellulose, so just cotton linters, we are able to sulfate that cellulose such that we can actually vary the degree of sulfation. And so that's a big finding. So again, if you look at the structure here, those R groups, we can substitute in those sulfate groups, and we can achieve a very relatively high degree of sulfation. And so what we found in some work then is that that promotes or helps to promote the sequestering of growth factors. And that's key, again, for presentation to the cells, and that promotes chondrogenesis. So I'll show some of that work. So varying that degree of sulfation improves biological kind of performance out of the materials. So shown here, we first investigated that, again, with the fully sulfated cellulose sulfate. We put that in media, culture media. We ran those cells in terms of forming these pellet cultures, and we noticed that the cells will become chondrocytes more readily in the presence of that fully sulfated cellulose. So we can perform histology, and shown down on the bottom there, those cells look much more, homogeneous and have that chondrocyte morphology in the presence of the cellulose sulfate and express the right markers. If we put them on the scaffolds now, the scaffolds that have that fully sulfated cellulose, we see a similar expression of that cartilage, these cartilage markers, high expression of collagen type 2, which is what you want to see. And you don't need much of it. So that fully sulfated cellulose, only about 0.1%, weight percent of that needs to be in those fibers in order to give you a high amount of collagen type 2 production out of those cells. Even in the presence or without the presence of growth factor, that TGF beta in the media, we also see this driving of chondrogenesis where the cells are expressing collagen type 2. So we look more carefully as well. We start to look at different amounts of sulfation in the cellulose and incorporate that into the fibers. What we found, interestingly, if you use a partially sulfated form of that cellulose, we see high amounts of growth in comparison to, say, scaffolds that contain that fully sulfated cellulose. And so shown here in those bars, that's numbers of cells, fold chains and numbers of cells over time. And so that's quite interesting. Again, for those defects where we want to promote growth of cells as well as differentiation, eventually that may be an important composition to use. It also will give you collagen type 2 production. So we look more carefully again at mechanism. As I mentioned, like I said, that degree of sulfation can affect protein interaction, and in particular, the TGF beta, which is a driver of chondrogenesis, that growth factor. So we see again with the fully sulfated cellulose, as shown here, that NACS, you get high amounts of that TGF beta interaction or binding in solutions, as well as on the scaffold. So incorporated into the scaffold. That's shown here as well. And what does that do? So we can run some additional experiments to look at the couple of things. So we wanted to, one, find out, do these GAG mimetics kind of outperform, right? Do these GAG mimetics outperform the native GAGs? Because why bother, right? I said back, a few slides back, you could just use the native GAGs like the chondroitin sulfate potentially, but we wanted to do something better. So does it really do better? Do the GAG mimetics actually do better than the native GAGs? So we compared our studies, ran studies where we looked at both native GAGs and chondroitin sulfate, different versions, C and A, there's different versions of chondroitin sulfate, as well as heparin. And so we look at collagen type 2 expression, which again is that protein found in cartilage that we want to see. All of them kind of produce pretty high levels of collagen. The cells produce a lot of collagen type 2 if you're on the normal GAG, the native GAGs, or on our GAG mimetics. But what's important to note, and that's what's circled there in the red, is that on the fully sulfated cellulose, you got less, or if not none, no collagen type 1. So again, with cartilage, you want to form homogeneous cartilage. That's all type 2 collagen production. You don't want the fibrocartilage, which contains that type 1 collagen. So the fully sulfated cellulose actually promotes that more homogeneous collagen type 2 matrix. So again, mechanistically, we wanted to look at that a little more carefully. And so, as we said, the TGF beta binds, right, or sequesters to these gamma mimetics. And so how do the cells sense that? So we looked at the expression of the TGF beta receptors. And so that's a receptor 2 and a receptor 1. So we did some relative gene expression. And so we noticed, again, high levels of that. Those genes for those receptors are expressed on those scaffolds that contain the gamma mimetics. We can look more carefully even at some of the other signaling associated with the binding of those receptors, which is the SMAD2/3 and phosphorylated SMAD2/3. And so we find that, again, if you have, again, high expression of that phosphorylated SMAD2/3, if you inhibit one of those receptors, only on those gamma mimetics do you get this kind of silencing down or lowering of that phosphorylated SMAD2/3. So it really shows that, again, the receptors, there's a direct link there. So sequestering TGF beta on those gamma mimetics promotes the TGF beta receptor interaction and then promotes the signaling, the SMAD2/3 signaling that then leads to chondrogenesis. Okay, so we've implanted those materials. And again, in these osteochondral defects in the rabbit, and we were able to demonstrate that that fully sulfated cellulose performed very, very well. So again, after 12 weeks in these medial condyle defects, we see full integration of the biomaterials actually resorbed by that time, but we see full restoration of the defect with cartilage as noted. Oops. Just click out of that. It's still going? Okay. So then, yeah, so we see here then at 12 weeks at the fully sulfated, we're getting that bridging of cartilage across, right, by that staining, the safranose staining, that orange that's shown there. And you get restoration of subchondral bone, which is that green stain. And so that's a mature, mature growth of cartilage tissue that you would like to see. The other materials, even the partially sulfated, was still at an earlier stage of repair. So very promising work. We're continuing those studies in different animal models to show that we can utilize this for our cartilage. And then we're currently also working on some variations of these gamma mimetics so that we can more closely represent some of those native gags. So again, if you look at the structure of the native gags shown here, right, they're really these, they have amine groups, they have different what we call glycosidic linkages, right, these beta and alphas, which is just kind of these orientations, right, in the structure. And so we wanted to try to look at that more carefully. We can create these gamma mimetics not only out of cellulose, but also starch and chitin, and those have those different types of linkages or functional groups. And so we've been showing, at least in some early data, that we are promoting chondrogenesis using those other types of gamma mimetics. And so this is exciting, and we're trying to see again where this will take us. Okay, and so now I'm going to switch to more of a physical property in our materials, and this is this phenomena of piezoelectricity, and we're utilizing this concept in both cartilage as well as in our neural tissue engineering work. And so I'll highlight some of the work that we're doing with the cartilage, and then move into the neural. And so again, piezoelectricity, this kind of electromechanical phenomena, as is shown here in this kind of cartoon. So when you squeeze on a material, push or stretch it, right, deform it. And that's kind of an exaggeration of the deformation. But when you do that, even minute deformations, it'll generate a charge, right? Kind of the surface charge, that phenomena. And so for decades, we've known that this is actually there in native tissue, bone, cartilage, it has this phenomena, as well as the native proteins that are present, the collagens, elastins, things like that. And so we would like to mimic that then again, with our biomaterials, we can create materials that have that same type of property. And the benefit here is really we can provide electrical stimulation, okay, into these materials with physiological movement. And so this is great. We don't have to apply external electrodes or other types of, physical stimuli like that, external to those defects. We can use the material, the implantable material in those tissues to provide the charge. And so electric fields have been shown to really affect growth and behavior. And I think the seminar speaker last week showed that type of response. So we are investigating this. We're looking carefully then at different materials, right? And so, like I said, your native proteins like collagen exhibit this type of phenomena. And shown here are some microscopy images that show this type of charging or deformation due to charge. So we use these model polymers, these fluoropolymers, these polyvinyl fluoride polymers. And what's nice about those is they're well-known biomaterials. They're used as sutures, okay? They are, used quite readily as sutures, but they are non-degradable, which is great for us. We can use them as a model. And they've been well characterized for their piezoelectric properties. And so when they're poled or in this all trans pole position or oriented dipole configuration as shown here, that gives it its piezoelectric properties. So you have those fluorine, you have those hydrogens. Again, when a movement is applied, you get this development or net polarization that occurs, development of charge. And so we can use electrospinning, our fiber forming process to actually pole them, which is also neat. So we performed a study, then looked at the effect of different processing, even post-processing of these materials, such that we can get a difference in that voltage output. So if you apply a load to these materials, you'll get either a lower or higher charge. And so we have these as spun formulation, or we have this annealed version. And so this annealed version, again, post-processing, we heat treat it, we are able to get a higher piezoelectric crystal formation that gives us a higher charge. And so shown down here, then we are getting voltages or fields, right, on the order of about 10 millivolts per millimeter. And on the higher annealed version, we're getting about almost on the order of 100 millivolts per millimeter, depending again on that frequency and deformation that we're applying. And so this is great. We can look at this and then now to see how does it affect our stem cells? Can we drive maybe chondrogenesis or even bone differentiation? So we use this bioreactor system. And so here we were able to apply a physiological load to these materials, dynamic compression at about 1 hertz frequency and about 10% deformation. And we looked at either as spun, what we call a low piezo, or a high piezo annealed version. And we compared it to our standard polycaprolactam control material. And so what we found was that our low piezoelectric materials, those as spun materials, gave us the chondrogenesis or formation of this cartilage tissue in those bioreactors about 28 days. So we see the nice staining of the safran proteoglycan, the type 2 collagen. We still had a little bit of collagen type 1 there, but not so bad, right, for 28 days, just in a bioreactor. And then our annealed one, surprisingly, no, right? The high piezoelectric, we got type 1 collagen, but we weren't getting type 2 collagen, right? So that low piezoelectric material promoted that chondrogenesis and showed with different markers as shown here. That higher piezoelectric drove more osteogenesis. So that type 1 collagen that we saw when you looked at some additional markers and mineralization, that was there. So that annealed higher piezo, okay, gives you that. And then we look at different markers, gene expression, the RUNX2, alk-phos, osteocalcin, all those things are expressed at really high levels on that high piezoelectric, the active material. So this allows us to do a few things. We can design our scaffolds now. Again, if we go back to our osteochondral defects, we can have the higher piezoelectric material sitting in the bone, the lower piezoelectric material sitting in the cartilage. So we can think of it from that. And we can also look at potential mechanisms for, again, designs that we may be thinking about that may be important as a next stage for cartilage regeneration. So the potential mechanisms, again, these electric fields that are generated are pretty similar in levels to what you see during early development. And mechanisms, because again, you're forming these surface charges, you're probably getting changes then in the surface of these materials. So we're looking at that more carefully. Adhesion of proteins, changes in local ion distribution can be affected. And then that's going to change, again, how the cells are going to be sensing that environment. So you may be getting calcium influx. Others are showing some of that. Again, electrical stimulation, you're getting this calcium influx, and that can alter kind of that whole mechanotransduction signaling. So we've used these materials now, again, as a model, because again, they don't degrade, which is great. So you can put these in culture and they'll stay, and you'll still have that piezoelectric property. And so we've used them in different systems. This is with the center, where we've got now looking at those materials as substrates for plant cells. Plant cells need a place to grow too in the lab, right? And so in these in vitro culture systems, it's nice to have a substrate for them. And so what's interesting to note is that cellulose does have piezoelectric behavior. So these scaffolds try to mimic that type of phenomenon of the plant cell wall. And so they readily will, plant cells will attach to these substrates. For us, we like to push forward with the development of these materials. Again, for application, we may not want to use those, non-degradable fluoride composition. And again, we're inserting these into the knee. And so eventually we would like ideally for this material to degrade, right? And you get the formation of the healthy cartilage tissue. So we've been looking at biodegradable versions of this. And as a first go round, we looked at incorporating or making composites that contain zinc oxide. Zinc oxide is well known to have also piezoelectric properties. It's been shown in other fields as these electric generators in various forms, but we can incorporate this into our fibers by, again, incorporating those nanoparticles into the fiber. And again, low concentrations of the zinc oxide into those fibers. We note that we can drive chondrogenesis. High concentrations of that zinc oxide in the fibers will drive more osteogenesis. And what's interesting about the zinc oxide, it will degrade eventually. So the zinc oxide will release, you'll get ion dissolution of zinc out of these scaffolds. And zinc is nice because it is an essential element in the human body. So it does other things, right? And it's also been shown to be an insulin mimetic. So not only are you getting piezoelectric properties from this composite, but you're also getting this release of zinc, which can be beneficial in other ways. We've been able to show then, so in these cultures, in this media, even without the use of additional growth factors, just using these composites, that you're getting, these cells are producing a number of different markers for both bone and cartilage. So you're seeing here the SOX9, collagen type 2, but you're also getting kind of secretion or production or gene expression for BMP2, which is going to promote bone formation as well. So it's interesting to see how that composite can affect the cells and driving them to differentiate. some of our newer work, and then this is going to lead into the, I'll finish up with some of our neural work. And so we've also, again, thinking about biodegradable versions of these scaffolds as our next iteration from the zinc oxide composite. We're looking at proteins, right? So we're actually going back to our gelatin scaffold, and we can make that piezoelectric, all right, by poling it. So gelatin is collagen, that native collagen. But when you process collagen in any way, collagen has in its native state, has piezoelectric properties. But when you kind of take it out of that native state, process it, it loses that property, right? So we can take collagen, denature it, form gelatin. Okay, it's going to lose that piezoelectric property, but we can put it back in and we can vary it, okay, improve the piezoelectric properties such that you can get high amounts of this piezoelectric output or lower amounts of piezoelectric output. So we take gelatin, we spin it, we form these fibers, we apply this high electric field to it, this poling. And we can vary this kind of the parameters on the poling. And then so then what results then is this change in electrical output. So I'm just going to put my arrow here. All right. So if we mechanically perturb those scaffolds, right, in tension for when we pole it, we're getting this about one, output of about 1 or so millivolts. In compression, we even see a much higher amount. So we're getting almost on the order of 30 millivolts out of these materials. When we partially pole, you don't necessarily in tension get that, but in the partially poled, you're getting some in the compression. We also see the unpoled, right? At least in the fiber form, we are actually still getting some charge out of that. And we can look at other piezoelectric parameters, these coefficients, that also give us an indicator of how piezoelectric these materials are. So this poled, again, D33 gives you a relative sense of, again, how charged these materials are. And so when you pole, again, you're going to have a side that may be positive and negative, okay, in terms of that polarization that you have. And so we get these D33 measurements that will be positive in charge or negative. And so that will become important when I jump into now and kind of finishing up a little bit into the neural application. We're just starting to do this work in utilizing these materials for the cartilage and bone area, where we really are seeing some exciting results is in the neural. So I'll jump back to that material in a second when I talk about kind of finishing up some of the work that we've been doing in the neural tissue engineering area. And so for neural applications, our interests are in spinal cord injury. We've been working with the group, University of Miami Project for some time now, where we are using these scaffolds as conduits to repair or regenerate those axons across those defects. So when you have an injury in the spinal cord, it can be very devastating, debilitating. So the idea is to use them to rebuild that damaged structure. And we've looked at our scaffolds, again, that same design and fiber structure, in whether in this case for spinal cord injury, we would like to get elong or extension of those axons as much as possible. And so we can design them either in a random form, random or aligned kind of oriented fiber arrangement. And this makes a big difference, right? So if you have nano-sized fibers or if you have larger micron-sized fiber, that orientation will result in to what extent you'll get this kind of axon growth or axon extension. So our kind of take home from this is that, again, you want to be on a larger scale, the fibers, micron-sized fibers, and you want alignment. And that helps to really get great extension of those axons. And others have also shown this, again, a number of studies, that this really is kind of key to be above a critical nano-sized dimension in order to achieve the greatest axon extension. So we've studied this now in a spinal cord injury model. And so we've looked at, in this case, Miami, the Miami project has been utilizing Schwann cells for many years. They've been studying for many years. They have performed clinical trials, phase 1 clinical trials, safety trials to show that those Schwann cells are safe and feasible to use in spinal cord injured patients. And so we're using Schwann cells in this case in combination with the scaffolds, and we find that they really do improve the overall extension of those axons or neurites, right? When we put those neurons on the scaffolds, you see a dramatic increase in neurite extension. I'm sorry I didn't give you a little bit of warning. There's a surgical image of that spinal cord injury model. It's a complete transection model that we've been utilizing to evaluate our materials. And here we implant those conduits, we add the Schwann cells, and what we find is that, again, on those materials, that piezoelectric, in this case, that fluoropolymer, you're getting robust kind of engraftment of those Schwann cells. The Schwann cells are the green. And you really don't see this, you don't see this kind of astrocyte or glial scar formation. Very little in that defect when you implant these conduits. And again, those Schwann cells help to promote that axon extension along those conduits. So we looked at whether or not we saw axons across that conduit, and we see that we get the greatest extension when we're using conduits that have the aligned fibers. And then also the astrocytes also were extending processes down those fibers and really help to promote the axon extension. And so I'll finish up a little bit, one more slide or 2 slides here, just to show, again, in those studies, we used the fluoropolymer, the non-degradable conduits, the piezoelectric conduits. And, and so again, for these injuries, ideally, we would want the material to go away or degrade. So we're going back to this gelatin, this protein-derived piezoelectric material that biodegrades. And again, we can alter the level of polling on this to achieve higher piezoelectric output in those materials. And so our recent work then shows here, and we're actually, my student Scott Strayton is presenting this at BMES. We are showing here that we are getting enhancement of neurite extension on those polled materials. And this is in a stretch device. So we're actually applying a cyclic stretch to those GRE neurons, 30 minutes every day for about 3 days. And so you can see, again, this P and PP are the polled materials that are generating this kind of electric output. And so a dramatic change here in neurite extension when you're using the piezoelectric, these more degradable piezoelectrics. And when you combine it with the Schwann cells, we also see that effect. But it's interesting to note, again, this degree or the orientation of those dipoles, that polling direction, right, that we're applying to those materials does have an effect. So it's really this polled negative or negative, right, surface that is giving us the greatest response in this type of system. So we're excited about these results. And we're moving forward with those protein kind of gelatin-based piezoelectric materials in future studies. So I'll finish up here. Again, the scaffolds. We've developed these scaffolds to really affect or have a biological response. And so we look specifically at architecture, that fiber dimension and orientation, but we're also looking at these physical properties, these properties of piezoelectric activity that will have an effect on response, and as well as composition, and really designing them well such that they will mimic that native ECM. So taking into account both physical as well as biochemical properties of that native ECM and driving a biological response. All right, so I'd like to acknowledge then my group, students that are, this is the most recent pictures of some of the work that was done in the studies. They're not there. They are now doing great things. But certainly acknowledge my current students, postdocs, and then as well as my collaborators, as well as funding sources. And I will take any questions that you may have. Thank you very much.
Thanks so much, Treena. And I'm pretty sure, you're looking for new student, new postdocs to join your new lab at Columbia here. So, so before I get into questions answered by Professor Lisa Olson, I'd like to want to call everyone's attention to a special edition of our seminar next Friday, September 30th at 11 a.m. Eastern Time. It will be our annual Van Mao Distinguished Lecture, and that's Van Mao is our funding chair of the department. So the lecture will be still delivered in hybrid format, but in our in-person audience, don't come here because the location will be at the Davis Auditorium. And don't come here, but I'm pretty sure some of you will come here. As a student, okay? We'll hear from Professor Celeste Nielsen as she presents uncovering mechanical forces that sculpt tissue for form. So please follow us on social media, receive a latest update. You can see our social media information on these slides. And you can always register information for the BME seminar at the website as well as YouTube as well. Now I turn to Professor Lisa Olson to answering the questions. Yeah,
Ask the question. So, yeah, that was a wonderful talk. I'm really interested in the PVD app. I have to talk to you about that afterwards. But so do we have any questions from students, the audience? Yeah.
Thanks for the talk. I was wondering, the sample size that you studied the adult stuff. Maybe also applying
Sure. Yeah, you can definitely. The IPS, we actually had a collaboration where they used the IPS cells and derived cardiomyocytes out of them. And so you can think of the obvious reason why you want to drive that with electrical charge. You can charge those cardiomyocytes, and that worked pretty well. We have a publication on that. So yeah, I think again, depending on the lineages that you're developing with those IPS or other stem cell types, they certainly could be utilized. Yep.
Great. So do we have any other questions from students? Yeah, great.
So do you ever work with kind of like structural heterogeneity in the constructs, and not just having one material that's the same composition, but testing multiple materials with different preparations?
Yeah.
Can you repeat the question?
Okay. Yeah. So you're asking whether or not we can have some heterogeneity. I can't speak in our materials. Meaning, I guess, like you can either have maybe different phases in those materials, they kind of bulk form, right? Or maybe even kind of gradient structures, right? Yeah. So we haven't done that yet, but it's certainly of interest. And I think because again, that we're finding that if we're starting to kind of use this term tune properties, and specifically in the piezoelectric materials, that you may want to have, again, these kind of higher electrical outputs somewhere, again, if I'm promoting bone formation, or lower output, like I said, for cartilage. And even with the cosaminoglycan, those GAB mimetics, we find that, again, some of them are promoting this growth, others really driving differentiation. So you may want to have those placed in different locations of your biomaterial if you're interfacing with other types of tissues. So yes, that's something we would like to do. We haven't done it yet.
So maybe we'll take one of the questions from the Q&A, and I think I'll start with the last one, which came in at the end. From Valentina Magno, very nice talk, Professor Arinzeh. Is that right? I was wondering if you have ever checked for the release kinetics of TGFB or other growth factors from the different sulfation variants of cellulose.
So we have, and so that paper is in revision right now where we're looked at sequestering as well as release of the TGF beta out of that fully sulfated cellulose versus say that partially sulfated. And so that fully sulfated version, like I said, will sequester large amounts of that TGF beta and it holds on to it. So it will release it at lower amounts than say that partially sulfated version. And so we've demonstrated that, and it will release over, a period of, we look maybe a week to 2 weeks out. We see those differences. Yeah.
Great. Are any other questions from students or anyone in the audience? Yeah.
Excellent talk, Professor. I just have a question. Has your lab ever looked at maybe how the materials that you're designing for defects, especially since you're interested in osteoarthritis, interact with kind of like an immune response over time?
Have we looked at whether they provoke an immune response? Is that what you're saying?
Yeah, or if maybe that they help like mitigate that.
Oh,
immune
mitigate.
response?
Yeah. So that's the next. So in that animal model where we had the osteochondral defect, we didn't look at that. We just, again, we're looking to screen to make sure our materials were safe, right? First and foremost, didn't cause any problems, but get that tissue repair. But the next phase of the work that we're doing, we're looking at that now to see if there's some changes in that kind of inflammatory environment where you're getting maybe a lowering of cytokines. Do the cytokines actually, because this stuff sequesters factors, well, it can sequester cytokines too. So we're looking at that, whether or not this material is doing something with those cytokines. And that may play a role here and why we're achieving repair. Maybe it's right sequestering and holding them so they're not available to those cells in that environment. So we hope that's the case. But we'll see, so we're running those studies. Yep.
So I think this question looks interesting by Oliver Coopers. Hello, and thank you for the great talk. Did you check the MSC differentiation capacity with and without the electrospun scaffold?
Yeah. So I think that question is whether or not are the fibers driving this phenomena, whether we looked at maybe films versus electrospun or fiber scaffolds. So some of our earlier work, yes, we've looked at films versus fibers. So depending on the system, the fibers do make a difference. The fibers do that additional topography, the physical cues, especially in the neurite, the axon work. Those axons are grabbing a hold of those fibers. And again, depending on the dimension, the axons are about 1 microns in size. So they are tracking along the fibers if they're of a similar dimension. If you don't have that, then you don't, like for films, you're not going to see that phenomena. You don't get that nice outgrowth. So yeah, yes, we looked at.
Do you, any other questions from students? I think we'll take one final question. So if anybody has one, but okay, maybe we'll do one final one from the online. Do you think one of those would be most interesting? Sorry, I don't.
Okay, let's see.
Better to choose.
Let's see. There's something more back. Maybe that first one, we haven't looked at that yet. What is the strength and integration of the bone at implant and after degradation? So I believe that is the composite material that we've developed, right? And so that composite, again, the ceramic is very brittle. We went to a composite so that we can have a more flexible material. And so the compressive kind of Young's modulus on that material is maybe on the order of about 100 kilopascals or so. It's lower than bone, but we're putting it into a bone and into a void, right? It's being used as a bone void filler. So there is, bone surrounding it, and it gets pushed into those defects. And so it does integrate well with the growing bone tissue. And so it resorbs over time. So we have not measured the mechanical properties of that regrowth bone after, weeks of implantation. But that's potentially important, again, important to do to get that. Information.
We've run out of time, the clock back there, so I think we'll thank our speaker once again. Everybody knows where we're gonna be next week, right? We all say it together, 1, 2, 3, David.
Yeah, thank you.